Oh my goodness, welcome back. My name is Darian.
My name is Aria.
And you are watching Night School, the live web series where we bring together scientists, artists, and creators to explore a new facet of our natural world every month and answer your questions. This Night School, 3 incredibly special guests are giving us an exclusive behind-the-scenes tour of a place seldom explored, the inside of a shark's nose.
A place that truly deserves so much more attention from the media, obviously.
True.
But hey, it's a new year and a new Night School, and starting now, all of the episodes of Night School will feature something even better than 3 experts, if you can imagine such a thing. This year, each episode we will be talking with 3 experts who are also collaborators, coworkers, and even friends. And they'll start off by giving individual lightning talks specific to their ranging expertises.
Then we'll bring them all together in the second half for a group Q&A where we'll hang out, we'll chat, and most importantly, we will answer your questions.
It's a science party. You're invited.
Now, when we connected with tonight's guests, we asked them what topic they'd want to craft an episode around. And I don't even think we were done asking we weren't able to finish the sentence. And they were like, well, we want to give a tour of a shark's nose, naturally. And so I knew at that moment that we struck gold. So we will begin our tour by exploring the myriad shapes a shark's nose can take. We see you, hammerheads. PhD candidate Amani Webber-Schultz will share how shape impacts a shark's sense of smell. Amani is a co-founder and the chief financial officer of Minorities in Shark Sciences, a nonprofit dedicated to fundraising for and creating a welcoming space for gender minorities of color in shark sciences. She also studies shark functional morphology and swimming kinematics in Dr. Brooke Flammang's lab at the New Jersey Institute of Technology.
And next, we are headed inside shark nostrils, where undergraduate researcher Gabi Castillo will share about the intricacies of the sensory structures that detect odors in the water. Gabi is a senior attending Florida Atlantic University, where she works with Dr. Lauren Eve Simonitis to expand our understanding of how different shark species use those internal structures to smell.
And to close out the night, we will hear about how it all comes together from Dr. Lauren Eve Simonitis, an aquatic sensory biologist who knows noses like no other. She studies the shapes, sensitivities, and water flows of different shark noses and how these features affect the way sharks interact with their environment. Lauren is a National Science Foundation postdoctoral research fellow in biology at Florida Atlantic University and the University of Washington, where she uses a bevy of biovisualization techniques to study sharks.
And the hype in the chat happening right now, absolutely. Yes, we are so excited about all of these guests.
I'm obsessed.
So excited to hear from them. And as always, you in the audience, let us know where you're watching from, whether you're with us on YouTube, Facebook, Instagram, or Twitch. Go ahead and comment, chat, share your questions, and we will collect all of your questions during the presentations to ask during the group Q&A session at the end.
Amazing. And with that, we will turn it over to Amani.
Hello, everyone. As they previously said, I'm Amani Webber-Schultz. I study shark functional morphology and fluid kinematics as they move through water. Specifically, I look at shark noses. So without further ado, sharks. Sharks have been around for more than 450 million years. They are an incredibly diverse group of species. They are older than trees and even Saturn's rings. And they have garnered quite a crazy personality when it comes to the different sharks that exist on this planet. And while they're also extremely well known, they're also very feared. And I'm sure many of you in the audience have seen this movie, Jaws, the OG shark movie. And one of the things that this movie really brought to life is this idea or concept that sharks can smell a drop of blood from a mile away. So for tonight, we're going to take you on a tour from the outside all the way inside a shark's nose and really break down this statement and give you the facts about how sharks actually smell. So shark noses, located right at the front of their face, just like with us, are very diverse. And when you look at the types of sharks that exist on this planet, more than 500 species total, they are diverse not just in where they live and what they eat, but also in what they look like. I specifically study dermal denticles, which are the scales found all across the body of sharks. They cover them from head all the way to tail, or nose to tail for the purposes of this talk. They are amazing in what they are made of. They have an outer enamel coating with dentine in the middle and a central pulp cavity, which might sound very familiar to you, and that's because it is. It is just like our teeth and a shark's tooth. In fact, the Latin translation of dermal denticle directly translates into skin teeth. So these skin teeth or scales were placed throughout the entire life of a shark, and I'm specifically interested in them because of how unique they look and this concept that they may or may not help with flow around the body of a shark. Now, these denticles layer very similar to if you were to pet your furry friend at home like a cat or a dog. If you move your hand from head to tail down a shark's body, it is smooth. But if you try to go from tail to head, it is quite rough. And these denticles are characterized by these crazy ridges, which you can see in this right-hand photo here. And they're also quite small, coming down all the way to around 100 microns in size, which is incredibly tiny and hard to see with your eyes. So shark heads in general are just as diverse as their bodies. If you think of something like a hammerhead with their crazy cephalofoil, then you have something like a nurse shark with such a blunt, big blockhead of a nose. Your tiger shark, which is just like a swimming square in the ocean, and even your really pointy black-nosed shark. All of these sharks have different nose shapes, but one thing that they do have in common is their nostrils or paired nares that they have on each side of their head. So if we flip a shark upside down, much like this illustration on the right, and we zoom in on its head, we can see these two paired nostrils or nares on each side of its head. And if we zoom in on just one, we'll see that there is an incurrent nares where research has theorized that water moves into the nostril, into that internal morphology where it can smell, and the excurrent nares where water then moves out. So what I'm really curious in learning is how these dermal denticles actually influence the ability of water to get into the nose. Because what place is more important than this sense that is so strong within us as people's idea of sharks, but also for a shark swimming in the open ocean, traveling long distances when they need to really be able to smell. Now, denticles, again, here on the right, they are beautiful and incredibly tiny. And one of the things that really fascinated me in trying to decide where I was going to study and then eventually landing on shark nostrils is that no other published papers have looked at this. Many of the published papers on dermal denticles have only looked at dermal denticles along the side of a body of a shark. So you might be wondering, how do we study something that is so small? I use 3 different methods. The first is CT scanning. Many of you have probably had a CT scan at some point in your life, just like in a hospital where you go into a CT scan. I sit at home putting sharks into CT scanners. This is simply a bunch of x-rays that you can stack into a kind of 3D volume that allows you to look at the thing that you scanned. I use scanning electron microscopy, where if you think of something like a typical camera, which uses light to capture an image, this amazing microscope actually uses electrons to capture an image and gives me really high resolution images of these very tiny dermal denticles. And lastly, I use 3D printing, usually resin printing, and something called particle image velocimetry, which is a complete mouthful, but essentially allows you to track the movement of a particle over a surface and see how that particle is acting. Much like if you put a canoe into a river to see where it might move along the river, if it would dodge logs or trees that were sticking up, same relative concept. So this is one of my lovely CT scans, and CT scanning is really awesome for giving you a large scale image of the thing that you're looking at, but what it doesn't do is give me a very close up image of the area of interest that I have. So in the CT scan, this hole that you have on the left-hand side is the incurrent nares, and the hole that you have on the right-hand side is your excurrent. And I'm really interested in that leading end of the incurrent nares. So our shark up here in the right-hand corner, it's showing you the orientation of the shark in the CT scan, because this is not the entire shark. This is a very tiny portion of it. And that incurrent nares right at the leading end of it is what I'm really interested in. So we have our little shark. We look really close at that incurrent nares. And one of the things that I found through my SEMs is this kind of insane patterning that you see looking at these denticles. So denticles layer from head to tail in a way that makes sense, in a way that makes it so sharks can move smoothly through water, often making absolutely no sound, and also reducing different physical mechanisms around them, such as drag. Now, looking at this image, I expected that all of the denticles would layer all the way down into the nostril, much like the denticles at the top of this image. But if you look closer, you'll see that there's these areas of convergence and divergence right at that beginning of the incurrent nares. And this was really interesting to me because the implication here is that fluid is doing something weird. Fluid is not just moving down into this hole. It is mixing, most likely, or maybe even separating out of the nose or coming up and out. So the arrows here represent fluid flow. You would expect to have water in that uppermost arrow moving from the head all the way down into the nares. What you would not expect is this arrow that is pointing in the opposite direction, this coming up and out of the incurrent nares. And this led us to believe that there was probably some sort of weird fluid mixing going on. We were really curious to see if this appeared on other species because, as I said, this has not been done on any published research. And so we looked at 9 species total, and we found this area of convergent and divergent denticles across all of these species, which was super fascinating. And because of this, what seems to be conservation of this weird patterning, the next thing we wondered was, what is flow doing? Can we even test it because of how tiny these denticles are? So our first hypothesis, and the one that we have run with, is this idea that these backwards denticles are creating mixing that is likely creating extra turbulence to remove larger particles out of the nose before they enter into that really sensitive internal morphology and tissue that you will hear about from Gabi in a second. This is very similar to something like a jet engine. So jet engines have areas of higher and lower turbulence, which allow for the removal of large particles that may clog the engine or harm the internal mechanism inside. But how the heck do we test this when it is so small? First, I take one of my CT scans and I have to build out an entire 3D model of it. This is hours of segmenting, roughly 3 weeks total if I were to work 40-hour weeks building this entire thing. I 3D print it using resin. We can then flip it upside down and lay it onto what I just essentially used was plexiglass and place it into what's called a flow tank. This is also sometimes called a fish treadmill, but it's basically a tank where you can decide the speed of water that you are moving over an object. So flipping this upside down, just to orient you, our shark's head would be on the left-hand side of the screen. And that area of interest, that area of convergence and divergence, is located right in this red box. Now, we had no clue what we were expecting to see. I was like, maybe this won't even work. Maybe there'll be nothing cool going on. Maybe I just wasted a year of my PhD doing absolutely nothing. And it turned out I was wrong. So this right here is what you get when you do PIV, or particle image velocimetry. What you see in here to orient you is this little red bit that we're really interested in is that area of convergent and divergent denticles. The white are particles that we've suspended in the water. So we've added particles large enough to appear when we shoot a laser sheet up onto the object that we're looking at, which illuminates all of the particles as they're moving. So you can see in the particles on the lower side of the screen, they're all moving at relatively the same rate. They're all moving in a laminar way, which means they're all moving relatively straight forward. And what I want to draw your attention to is that region of interest. And as you stare at this long enough, you'll notice that there appears to be some weird little mixing where the particles in that area are not only slowing down, but also appear to be moving not in a laminar or straightforward way. So when you add your vectors over this, these green arrows represent just the relative direction of the particles that they're moving and also the speed that they're moving at. And you can still see that kind of laminar straightforward flow. The area that we are looking at, they look like they are going in a little bit of a vortex area, which made me really excited. But one of the things that's really hard to see from this is what those arrows are actually doing. So when you take a still and you track the motion of water over this, you can see this beautiful vortex, which is exactly what we were expecting to see. Now, this is really interesting because the implication for this is that sharks have developed a way to mix the water before entering into their nose, which has big implications for the ability to detect an odor and capture odors as it's entering into their nose. And as someone who did not previously study smell and only smelled denticles, being able to team up with these two amazing speakers after me was really exciting because it meant I could not only learn more, but I could kind of mesh our two worlds together and create a very holistic view of what's going on inside of a shark's nose, especially when we don't really talk about it that much. So upon all of this inspection, we not only figured out that the dermal denticles look absolutely insane at the incurrent nares, which was completely not expected, but they also appear to be doing exactly what our hypothesis thought, which is incredibly exciting. And I am so excited to continue to move forward with this research, especially with these two speakers after. So this is what's occurring on the outside of a shark's nose. But what happens on the inside? with that, I'm going to pass it on to the next two speakers.
Amazing. Thank you, Amani, for introducing us to the outside of a shark's nose. Cannot wait to get in there. But first, brief question for you. What was the first shark that made you fall in love with shark science and shark research? What was your gateway shark?
I would be lying if I didn't say it was a nurse shark. It was the first shark I ever touched. And if you know shark researchers, you know that shark researchers think nurse sharks are insane, but I have a special place for them in my heart.
Okay. I'm gonna put a pin in the question of why that's the case for group Q&A, but yes. That's a great answer. Nurse shark, thank you so much for sharing. And we're gonna go ahead and pass it on to Gabi.
Okay. Hello, everyone. Good night. My name is Gabi Castillo, and tonight we will be taking a closer sniff at shark noses. So just a little bit about me. I am a senior studying biology at Florida Atlantic University. I'm currently studying at FAU's Harbor Branch Oceanographic Institute up in Fort Pierce, Florida. A part of the Semester by the Sea program. I am a member of the Minorities in Shark Science nonprofit, and it's an amazing organization that helps racial and gender minorities get kind of make a breakthrough in this field of shark biology. And I also work with Dr. Eve Simonitis in the FAU High School Imaging Lab, where we are studying and taking an inside look at what the internal morphology of different species of sharks olfactory systems look like, particularly through using scanning electron microscopy. So sharks can't sniff. So how exactly do they smell? So as Amani kind of walked us through, sharks, instead of being able to sniff like us, they cannot intentionally sniff. So their ability to smell is not linked to respiration or breathing like ours is. So instead, as sharks swim, they pick up dissolved odorants in the water in order to detect different scents, which is why they're often referred to as swimming noses. So as Amani kind of talked about, sharks' external morphologies, including their nares, are absolutely crucial to their ability to smell. So as sharks swim, water will passively flow in through their incurrent nares, their nostril or opening of their nose, which will lead into their internal structure of the olfactory system. And as you can see at the image below, with variation in species, there's great variation between their external olfactory morphologies. And we see a similar difference between the internal morphologies of sharks' olfactory systems as well. And olfactory has to do with essentially anything with the ability to smell. So this schematic overview, it's of a bonnethead shark, and it shows that internal olfactory system. So as water flows in through that incurrent nares, it passes over an organ called the olfactory rosette, which is comprised of a bunch of different individual lamellae, which are like these thin sheets or layers of tissue. And these rosettes are housed within a cartilaginous olfactory capsule. And they're also attached to something called the olfactory bulb, which is actually a part of the shark's brain that has been moved forward to connect to this rosette. And the bulb connects to the actual forebrain or telencephalon via way of the olfactory peduncle. So as Amani kind of mentioned, we can view the internal morphologies of different sharks' noses through CT imaging. So here we're depicting a CT scan of a bonnethead shark. And on the right, you can see the exterior olfactory system of a shark. In particular, take notice of that nare or nostril. And then on the left, we see what is the internal morphology. So as water flows in through that incurrent nare, it passes over the olfactory rosette, which is shown in purple. And you can also see individual lines within that rosette that are the shark's lamellae. And once again, once water flows over this rosette and passes over the lamellae, it connects into the telencephalon if any dissolved odorants or chemicals were absorbed along this lamellae, passes that information onto the brain for further processing. So when we start to look at different shark species, we can see great variation in these internal olfactory morphologies. And two great species to look at are the Pacific spiny dogfish and the bonnethead shark. And obviously these sharks have significant differences within their external snout morphologies. So the Pacific spiny dogfish has a much narrower head, while the bonnethead shark, it's a member of the hammerhead family, so it has that kind of elongated horizontally cephalofoil. So obviously their external morphologies are very different. And when we compare their internal morphologies, we see great differences as well. So looking back at that CT scan, we can take a closer look at that olfactory rosette of a bonnethead shark. And we notice this is a pretty relatively long rosette, and it appears almost kind of like a lunar phase. And when we an even closer look at that rosette, we can notice the formation of the top of every single individual lamellae, which make up that rosette organ. And when we compare a CT scan of the bonnethead shark, shown on the left, to that of the Pacific spiny dogfish shark, shown on the right, we see significant differences between their rosette shapes. So again, bonnethead sharks tend to have that much longer, kind of lunar-looking rosette shape, while the Pacific spiny dogfish, with this much narrower head, has a much more almost circular, bulbous-shaped rosette. So with these obvious differences in head shapes or external morphologies, we see differences as well in the rosette shapes between these two species' olfactory systems. So if we again look back to that bonnethead rosette, we can essentially take what is a cross section of this rosette, or basically if we were to have it. And if we took this half and we rotate it on its side, we can see what is the face of that is a singular lamella that again are those thin sheets that make up this whole rosette. And with identifying this, the lamella are essential to the function of the whole system as a whole, because each individual lamella significantly increases the surface area that allows for better absorption of those dissolved odorants and chemicals within the water that allow sharks to detect different scents. So on the left here, we see a drawing of what is an individual lamella, and they appear almost like butterfly wings or angel wings, which is kind of cool. We can see that point of attachment to that olfactory bulb, which is again part of the shark's brain actually, as well as that inner membrane where we see some cool trends, which we'll get into in just a minute. In the middle, we can see images of 21 different species of sharks' individual lamella. So we can see great variation just between lamella size, even almost color. And on the far right, we see what are two individual lamella from the Pacific spiny dogfish shark, which is particularly what I look at in Dr. Lauren's lab. And these lamella have undergone dehydration through a critical point dryer, but this is right before they receive a sputter coating, which is of a filament that's a gold palladium filament that allows us to actually see them when we put them under a scanning electron microscope. And with SEM imaging, what we can do is we can see that within each of these individual lamella, there are different regions of where we see sensory epithelial tissue as well as non-sensory epithelial tissue. So how exactly can we spot the difference between sensory and non-sensory regions? Well, it's actually fairly easy. So sensory tissue, something that Dr. Lauren told me when I just started out in her lab that has really helped me and that I still use, is that sensory tissue looks exactly like spaghetti. plain and simple. And then regions of non-sensory tissue, they don't look like spaghetti. And instead, they actually appear to look somewhat like honeycomb in that they're almost hexagonal, but not really. So if we were to take a look at a transition zone between that sensory and non-sensory epithelium, we can see here on both images where that spaghetti-like, heavily ciliated sensory tissue is. And it's on the upper portion as well, so you can't tell. And just on the lower left part, you can see that non-sensory tissue, which is much smoother in appearance. And it has that almost hexagonal, slightly honeycomb, almost cloudy formation looking. Which is pretty cool. And we do see occurrences where areas of non-sensory tissue actually appear to protrude within non-sensory, which is interesting. And then if we look at different species of sharks, we can see not only differences in their rosette shape, which we kind of touched on with differences in bonnet heads and Pacific spiny dogfish, but we can also see differences in their actual bulb sizes. And again, bulbs are important to their whole olfactory system as a whole because they are a part of their brain. So in determining the differences in bulb size, we actually learn a little bit about how a species of shark relies on olfaction as a sense. So here we see the olfactory system of an Atlantic stingray, so a ray, and we can see the telencephalon, again, the forebrain, and how it connects to the olfactory rosette depicted kind of in that dark color all the way on the right via the olfactory tract or peduncle. And we can see that the rosette kind of has like a C shape to it, and that it is directly attached to the olfactory bulb, which is rather thick for a species of shark, and that, again, it has that a thicker olfactory tract. So when we look at a different species of elasmobranch or shark, if we look at the lemon shark in particular, we see once again the same olfactory organs. However, if you look at those rosettes, once again, the olfactory organ is what they're labeled as here. They appear a lot straighter. And interestingly here, we see a difference in the bulb from the Atlantic stingray. So lemon sharks actually have what is called a hemi-olfactory bulb, where there's almost a divide between each of the bulbs into two different sections. And if you also note at the olfactory tract or peduncle, it seems a lot narrower than that of the Atlantic stingray. So again, we're seeing a lot of variation just between two different species of elasmobranch. And then if we were to look at another shark, the bonnethead shark, for example, we see even more variation. And again, it has the same olfactory organs that the other two elasmobranch species do. But again, what's unique about the bonnethead shark is that they have that kind of horizontally elongated cephalofoil. And this changes their internal morphology as well. So their olfactory rosettes appear almost lunar shaped. And if you look at their bulbs, at the point of attachment to the rosettes, they appear a lot thinner than the other two species, and the tract itself is also relatively thin. So again, this kind of just highlights how we see a similar trend to external morphologies, how they differ from species to species, and how when you look at the internal morphologies of different species of sharks, we see great variation there as well. And a study conducted by Dr. Jopak in Australia back in 2015 kind of compared these differences in internal morphologies of the olfactory structures of different species of sharks. And what they particularly were looking at was differences in bulb sizes here. And what these researchers found was that differences in bulb size correlate with differences of a shark's role in ecology. So basically, the type of environments that a shark inhabits plays a significant role into determining how much a species of shark relies on their sense of olfaction or smell. And we can tell sort of roughly how much a shark relies on its sense of smell by analyzing their shark bulbs. So a species of shark that has a large olfactory bulb, we can basically assume that they rely quite heavily on olfaction as one of their main senses. And what Dr. Jopak found from her study was that the largest bulb sizes can be found in species that are oceanic or pelagic sharks, since they don't rely as much on their vision. So these bulbs tend to be somewhat increased in size. And there's a similar trend with deep sea sharks as well, in their olfactory bulbs also being quite larger, which we see similar in deep sea bony fish as well. And then the smallest olfactory bulbs were present in sharks that typically inhabit reefs. And this is really due to the reef environment, since there are so many different species and there's so much going on, both not just smell-wise, but also in sight. Sharks that inhabit reefs tend to kind of evenly use their different senses. So their bulb size are slightly more proportional and not as large as we would see in those deep and open ocean species of sharks. So now that we know a bit about how what the internal morphology of these different sharks looks like, I want to turn to Dr. Lauren to find out how exactly they work and how water flows through them.
I am also so excited, Gabi, to hear how they work. Thank you so much for that amazing presentation. Before we let you go for a second, I do have to ask you the same question Aria asked Amani earlier. What was your gateway shark that sparked this passion for you?
Definitely a bull shark, because I was kind of raised in North Carolina, where we lived in lakes. And I remember just hearing as a kid on Shark Week that sharks could survive in fresh water. And that actually scared the living hell out of me, because I was convinced as a kid that they were in the lake. So they're euryhalene, they're very cool. So that's my gateway shark.
Amazing. Thank you. We will hear back from you in just a second, but now I would like to turn it over to Dr. Lauren Simonitis.
Hello. Awesome. Thank you guys so much for tuning in and hearing about shark noses, which clearly are one of my favorite things to talk about and do not get enough attention. So I'm so glad for this platform to bring it to you all. So just a quick thing about myself. I love looking at noses. I love sensory morphology in general, but I love noses, and I love looking at them across the board on any fish without bones. So I mostly look at sharks. I look at some rays, some skates, their flat cousins, but also some of their more ancient looking cousins like this chimera over here. Today, we're really going to focus in on sharks because I could talk about this for the whole 45 minutes, but we'll just cue in on some of the shark species that we look at and kind of tying together both what Amani and what Gabi talked about earlier. So one of the things that I thought about, because I spend a lot of time, a lot of people think shark researchers are out on the water catching sharks. And I can say that's probably 2-3 weeks out of my year. What I actually do is I spend a lot of time on computers and microscopes. And I used to think that I looked at shark noses all the time and was looking at really cool sensory morphology. And I actually work in a K-12, which houses my lab. And one day, a couple of students were coming through giving a tour, and they were like, "Oh, is that a shark booger? Is all you do look at shark boogers all day?" And I was like, actually, yes, that is much more fun to say that I do. I look at shark boogers all the time. And basically, I try to look around the shark boogers and look at what is doing the sensation within the nose. And you guys have seen this photo a couple times. I think it's beautiful, so I'm always happy to look at it again. But this is that bonnet-fed olfactory system. And if we think about how water is flowing, we've heard kind of the general pattern of how water goes into the nose. But with this CT scan, I'm going to show you some different those actual cavities where water is flowing through and how water actually is getting into this nose, carrying those chemicals that the shark is smelling and responding to. So as you guys probably know, CT scans, like CAT scans, like Amani and Gabi both touched on, are a bunch of x-rays stacked together. And if you know anything about sharks, they don't have bones. They're actually overall pretty squishy. They have some hard parts. So if you put a shark into a CT scanner, you're not going to see too much. You're going to see some hard little scaly bits like Amani talked about, some of their cartilage, their teeth, but you're not going to see any of the soft bits, which is what I'm super interested in. So what we do is we put them into this dye, this radio-opaque dye that all of their soft tissues take up, and that makes them radio-dense, so that when we shoot x-rays through them, we can see all these different soft tissues we wouldn't normally see. And that lets us not only see the tissues, but see these really important cavities. So we've talked a little bit about the in-current nostril and the in-current chamber. So this right here is terminating in the in-current nostril, and this whole chamber right here is the in-current chamber. So water is flowing in through this in-current nostril and making its way through this chamber. So then if we look at this next chamber out here, this excurrent chamber, the water is flowing out here and coming out of this excurrent nostril. So it's this unidirectional pathway, or so we thought until Amani found all of her cool stuff. So we're still trying to understand and investigate how water is flowing through these structures. And we've looked at this too. We've looked at this 3D structure of this beautiful rosette. We've looked at that lamella, and when we've looked really close, we can see this differentiation of sensory area versus non-sensory area. So if you can kind of see, the sensory area has these secondary foldings. These increase that surface area so that more water can come through and can bind through to more sensory little receptors, which are called the olfactory receptor neurons. And we can kind of divide this into sensory area versus non-sensory area. And again, we can see this spaghetti versus this honeycomb shape. And what Gabi didn't get to yet is my favorite part of the spaghetti, which if you look through here, I like to find the meatballs. And the meatballs in the spaghetti are right there. And if we blow that up, that is an olfactory receptor neuron. So basically, they have little microvillus endings. And all of these ciliated cells, the spaghetti around it, they beat and they move mucus across this cell. They move shark boogers. So shark boogers are really important because just like in our nose where we produce mucus, what happens is mucus is really, really good at trapping chemicals. So water is rushing by with chemicals. And basically what happens is as water is passing by with chemicals, the mucus grabs onto the chemicals and helps bring it to the olfactory receptor neuron. And if we think about how that works conductively, like through the system, again, we have, if we're looking at this little guy, so this is our meatball within the spaghetti, and we have water coming through with chemicals. The chemical stays on, binds to that end of the olfactory receptor neuron. The olfactory receptor neuron sends an electrical signal. It's called an action potential. And this signal is saying, hey, I have attached to this chemical. I am smelling something. And that signal travels up through the olfactory bulb and then through that peduncle and through the brain, which Gabi took us through so nicely. And if we want to look at that signal again, water is coming through. It's binding to that olfactory receptor neuron on the lamella. And that signal is going up from the olfactory receptors to the olfactory lamella through the olfactory bulb, peduncle, and brain. So from there, the shark can then be like, all right, what do I want to do about this scent that I'm smelling? Sharks are smart. And that's something that I think people don't really get, is that sharks have a variety of systems. They are not just smelling a drop of blood and going into a frenzy all the time. It does happen, but there's been plenty of times where I have had sharks and I give them a little bit of blood and they're like, you know what? I'm actually not hungry today. So that smells okay, but I'm super full and I don't want it. So sharks are able to make decisions based on the cues that they receive from their environment. And one of those is smell. And from what we know, about how water flows through a nose, we know almost nothing. So this is a paper that came out and it is great. It is one of the only really in-depth studies of how water flows through a shark's nose. And this is using something called computational fluid dynamics, where basically they model how water would flow through these chambers. So this is water coming in through that in-current nostril, going through that in-current chamber, coming back out this excurrent chamber, and then out this excurrent nostril. This is on a small eye hammerhead, and you will see that it kind of looks very similar to the bonnet head olfactory system. So we only know it on this really long, elongated, really dramatic shape of a nose. And what they found was that there were differences along this rosette. So they found that there were areas that had really high flow, and that's kind of happening in the middle of the rosette. But if we go to areas that are closer to the nostril, like over here, these little dark blue areas here, or this big dark blue area here that's close to the center of the head, we see very low flows. They actually called it in the paper a near stagnant recirculating area. So when this paper came out, I was like, okay, I have a very similar nose that I look at. I look at the bonnet head shark nose all the time. Do we see any differences in the lamella the individual little stacked dishes in a dishwasher, like I like to call them, if I took each of those dishes out and I looked at them, would they look different based on where they are in this nose since they're experiencing different flows? So that's what I did, is I looked at how much area of each lamella at each position along this along this rosette, how much of the area of the lamella is devoted to sensitivity, how much of that is covered by that sensory epithelium. And what we found was that the places in the middle that had this high flow, they had higher sensory area. So more of their area was devoted to sensitivity, and they had more of those secondary folds that increases surface area. But then if we looked at these blue areas, they had low sensory surface area. So this kind of made us think that the flow is correlated with sensation. So areas that have low flow have less sensory epithelium than areas that have high flow. And this kind of really got us thinking about, okay, this is just one type of shark, and it's a really dramatic, elongated shark. So what about with different noses? There's so many different noses. We have sharks in all shapes and sizes, and their noses are really different too, as we've been over. So let's look at a bunch of different sharks, try to see, do their noses follow the same pattern, and can we see how water flows within the nose? So I've been looking at just a ton of shark heads. I have amazing collaborators all over that will either send me scans or will send me shark heads in jars. So I have a very creepy mad scientist office. But I pop these guys into the CT scanner and look at them in the scanning electron microscope and try to figure out what is going on within their noses. And one of the most exciting guys, especially when we think about sensitivity and flow, is the Atlantic shark nose. And nose is in their name, which should have been a giveaway. But honestly, these guys are kind of regarded as a lame, trash shark, because whenever we take students out and we take them out shark tagging, they want the great hammerheads, or they want the tiger sharks, or the bull sharks, or all these giant sharks. These guys are pretty small. And whenever we get them, everybody's really excited to see what shark is coming up, and then they see it's a sharp nose and they're like, oh, okay, well, we're gonna go sit down. They're not excited about it. But I found that these guys are super exciting. So just to kind of, reiterate, going through how this shark nose works, when we push through this shark nose, we see those same patterns. So water is coming in through that in-current nostril, going in through that in-current chamber. It's then going to turn right back around and come out that ex-current chamber, out that ex-current nostril. So we can see that the patterning that we understand is the same, and it looks very similar, right? There's still those stacked dishes in the dishwasher that make up that rosette. But what we found interesting was when we started going through this scan, how we make this stain that we put them in that makes them nice and dense so that we can see, is we mix this powder with water and then we put them in this stain. And the issue is if you don't mix the stain well, sometimes it you get little clumps of powder, or if it gets old, if it comes out of solution, you got to make new powder. So I was looking through this scan, and I was we're going to be going as if you're flying through the nose. So here's the lamella. We're going to be going through the nose. And what you're going to see is kind of this little light show. So there were all of these little white dots showing up. And I was like, well, what are these white hard parts in this nose? This should not be here. Noses are very squishy. We all know this. Those of us who work with noses, which is 5 people, we know that they're squishy. So why are we getting these nice little hard parts all scattered through? Is it powder that has come out of solution? What's gone on? Did I mess up my scan? And when I put it under the SEM, I found something that was shocking. And that is these weird structures that you're going to see down here. But first, to kind of remind you of where water is flowing, it is flowing through this in-current chamber right here, right through this lamella. And then it's going to turn around and come back out this ex-current chamber down here. Okay, so when we look at this area here, you see these fields of little spikies. And when you get close, there are these leaves. And I was shocked. I texted a lot of my friends who work on sharks, and I was like, I have leaves in a nose. What is going on? Also, just to show you, this is how small they are. This is 200 microns. That's 200 micrometers. That is 10 to the -6, right? Tiny, tiny, tiny. And so you can't see them by the naked eye. I was like, what's going on? I think I texted Amani, she's denticle queen. And I was like, are these denticles? They look like denticles. They're ridged. And yeah, when we got close, we saw these are indeed denticles. And they have these ridges that helped direct flow. And when we think about where water is flowing in this nose, it makes sense. So these denticles are all pointing in the correct way where water is flowing. But then we had this whole patch of denticles up here that are indicated by this orange arrow that are oriented opposite. So are they doing that jet propulsion thing that Amani has theorized? Are they sending water back to be resampled? What is going on here? And then it got even weirder, because if I look away from this big channel, I keep seeing these spikies, and I see them in this area that they create these little claw shapes at the end of these secondary folds. And they're all kind of pointing different directions. So I don't really understand how that helps water flow. I mean, I guess they're kind of pointing in the correct way. We also have them in the center of these secondary folds. And now I want to remind you too that normally secondary folds are completely covered by sensory epithelium. So they're completely covered by spaghetti. But these guys are covered in non-sensory epithelium. So they're actually sacrificing area that is normally used for smelling to put denticles in, to put these water directing structures in. So that really speaks to how important these structures are, because they're sacrificing areas that they can smell in order to put these denticles in. This is also like the only shark I've looked at so many papers. This is the only shark I've seen this reported in. I haven't been able to see this in any of the other heads that I've looked at, any of the noses I've looked at. So just really weird little guys. And again, these are these little spiky bits coming off of these secondary folds. They're kind of going in the direction that we think water should be flowing, because water not only is coming out here and out here, but it should also be flowing within the lamella. So they are kind of pointing as like flowing down with the lamella, but it's still just weird. And they are like fully embedded. At first I thought maybe I had like nicked them on the outside and brought them in, but no, they are like fully part of this structure. And this just really goes to show that there is still so much that nobody knows about these shark noses. There's very few of us working on this. You've met like half of the team tonight. So this is a wide open field. And I just want to really encourage people to think outside the box when they think about shark science, and think about all of the things that we still don't know. So there's so many more areas of shark science than you see popularized on the media or on popular shark shows. There's so much left to know. And a lot of the opportunities that we get to study these kinds of things are with Minorities in Shark Sciences, which I know has been mentioned tonight. You can scan that QR code if you want to learn some more about us. But we are, like has been mentioned, we are a group of gender minorities and racial minorities who are in shark sciences. We provide a lot of opportunities for our members, both virtually and in person. We just had our first annual summit. It was a huge success. It was a huge amount of fun. And we got some cool new sensory projects starting up. So there's work going on in the sensory world, but we really need help. So if you're interested in shark science, and this is really cool, it's wide open, and I cannot look at all these heads by myself. So please help me. And with that, we have a lot of people to thank. This is kind of like our group thing. Thank you guys so much for having us. And thank you to everybody that has supported us, given us time. And
oh, I think we got Lauren frozen there for a second, but you heard it from her first. Go check out Miss, really amazing organization, tons of amazing opportunities, and super excited to hear that this first summit went well. And after Lauren comes back, we're going to go ahead and bring everybody else back up on the screen for a little group Q&A session. So anybody who's watching...
Sorry, that was my bad. Transition too soon.
That's okay. We'll ask Lauren the very important question of Gateway Shark when she gets back. But hi, Gabi. Hi, Amani. Welcome.
Hi.
Thanks so much for joining us again tonight. Yeah, go ahead, Darian.
Yeah, this has been amazing. I've so enjoyed listening to all of your talks. I'm honored to have 3 of the 5 shark nose scientists, according to Lauren, in the room with us tonight.
Yeah, welcome. You're in the inner circle now, because this is it.
Oh my gosh, the inner olfactory bulb, maybe.
That was a good one.
That was good.
Thanks, I learned it from y'all.
Awesome.
Great. Well, so we have a lot of really great questions coming in from the audience. Keep asking them if you're watching out there. But getting right into this first one, how did all of you meet? Say more. We know a little bit, but what's the story?
Oh goodness. Well, I just met Gabi a month ago.
Yeah.
Lauren, let's see. I met Lauren through my nonprofit Miss, and then I met her when I did a really fun fish morphology class at the University of Washington in 2022, where she kind of dragged me willingly into the area of shark noses because I came into that class really wanting to do dermal denticles, but I didn't know where. And she was like, "What about a nose? What about nostrils?" And I was like, "Sure, I'll pick some noses. Why not?" And that's how I met Lauren. Gabi, do you want to talk about it?
Yeah. So I actually met Dr. Lauren through a mutual. Her name is Deborah Santos de Azevedo, and I met her on actually I met her twin sister in a coffee shop anyways, and she told me about Miss, and I had the opportunity to go out on a weekend-long shark survey, and I've done some of Deb's personal surveys, which is how I met Lauren, and she was like, if you ever want to get involved in shark research, and particularly nose research, I was like, hell yeah. So yeah. And then I met Amani like a month ago over email. So yeah.
Amazing.
I know Lauren shared that QR code of how to get involved with Miss, but if there are any like potential young shark scientists in our audience tonight, like how would you recommend that they like start in this field?
Oh goodness. Honestly, I think just be curious about anything. I did not expect to end up studying sharks. I kind of fell into them close to the end of my junior year of college. I spent a lot of time working in a lot of other marine science labs, and I kind of just wound up doing shark things. So I would say just be curious and work really hard in the areas that you end up with opportunities. And I'm a very firm believer that if you put things out into the universe that you want, they'll eventually kind of find their way to you. So that's my recommendation.
Awesome. All of you work with CT scans at sharks. We had a lot of comments across platforms. On Instagram, I was seeing them pop up on YouTube. How the heck do you get a shark in a CT scanner?
So I dissect the noses off of sharks. So Lauren said she's creepy lady with shark heads. I'm the weirdo who DMs people when they say a shark washes up in a state somewhere, and I'm like in the Fish and Wildlife's DMs being like, "Can you cut the nostrils off for me? Can I have those?" And so I have a creepy little jar of shark nostrils. But you can also put a full dead shark into a CT scanner. So museums are amazing in that they keep specimens, so they will have full 6-foot sharks even. And I know some people who have gone to hospitals and put sharks into human CT scanners.
Do Fish and Wildlife like know you at this point? They're like, yeah, Amani, just like take whatever you can.
Probably. I'm sure they've heard of me if I haven't talked to them already.
Oh, that's so excellent.
Lauren, welcome back. Sorry you got cut out for a second. We do need to know what your gateway shark was. I don't think we're going to let that question fly.
Yeah, it was like the worst time for my Wi-Fi to just stop.
It was perfect at the end.
I was not really a shark person until the last year of the couple last years of my PhD. I was studying sea slugs, and I was studying how sharks reacted to sea slug deterrence. So I just found bonnet heads because I was like, oh, their olfactory system is huge, and they're so cute and easy to have in an aquarium and test. So bonnet heads were my gateway that I was like, oh, wait, sharks are actually very cool.
That's an excellent gateway shark story. Starting with the slugs and ending up at the sharks. Love it.
Yeah, I'm still a slug girl at heart, but yeah.
Slug girl at heart. Can we get that in a sticker? Someone out there, please. Amazing. Well, as we were saying, we have a lot of great questions from the audience, and another one that's come up is the nostril the only way for scents to enter a shark? Is there any water flow from the mouth? Kind of like how human taste and smell are connected. This is a question for all of you. So
yeah, so sharks only really smell with their noses, and they don't have any nose to mouth direct connections unless you look at chimeras, ratfish, which are like these distant relatives of sharks. I showed them at the beginning of mine. They kind of look like ancient sharks. They have a nose to mouth connection, which is really interesting. But also sharks have something called the solitary chemosensory cells. It's just the general chemical sense. And they're just kind of like scattered on their skin, but they only respond to like distasteful stuff. So they're kind of known to like respond to like nasty smelling things.
How does that kind of nose respond to like what is considered nasty smelling things?
Yeah, like we think of like bitter, or like they respond like that is what things have like they've isolated as being responsible for responding to a lot of the like the things that cause sharks to freak out and like not smell good. So like chemical deterrents that animals make. But we know even less about that than we know about noses, and even less than tongues, because we know very little about tongues.
So the order is tongues, noses, deterrents. Is that what I'm hearing?
Noses, tongues, solitary chemosensory cells.
Okay.
Just a whole mystery.
Shark scientists
Full
have, like, you have like the best vocabulary, like the best words. Like Gabi, every time you said peduncle, I was like, I would not be able to make it. Like peduncle is one of the funniest words ever invented. It's just such great vocab in shark science. Speaking of sharks' relatives, the great mistake on Instagram wants to know, are sharks and rays related / are they friends? Do rays have denticles too? So I guess that's 3 questions. 1, are sharks and rays related? 2, are sharks and rays friends? 3, do rays also have denticles?
Go for it, Amani.
Okay. So sharks and rays are related. They fall into elasmobranchs, which is sharks, rays, and skates. And that kind of falls into the very large group chondrichthyans, which is just cartilaginous fishes. So in that you have your chimeras, your sharks, your rays, and your skates. And your sharks and rays are kind of closely related to each other in that kind of evolutionary history. Rays do have denticles. They don't cover their entire body though. So they can be found in very specific regions. And they do not kind of overlap in that crazy way that you see sharks do. So sea pancakes are in fact just like soft, soggy mushrooms with like random spikes on their bodies.
Sorry, sea pancakes? Say more.
This is indeed what I call rays. I call them sea pancakes.
Okay, excellent. So that's you heard it from a scientist. Sea
Yep,
pancakes.
straight from the scientist. So we'll put that, tell Webster Dictionary.
Perfect. Get him on the phone. Oh, excellent. And we kind of visited this a little bit earlier, but we're curious to hear from all of you, why shark noses, or why noses generally? What really drew you to studying them?
That's Lauren's fault.
I'm just like preaching the shark nose gospel everywhere I go. And then because Amani had come into this class that I was teaching, and she wanted to study denticles on some other part of the body, which everybody looks at. And I was like, we're
calling me out. Wow.
Nose.
I drank the Kool-Aid. It's true. I drank the nose Kool-Aid.
The nose Kool-Aid.
The nose Kool-Aid.
I think it's great how, like, Lauren, you're an influencer.
Oh my gosh. That's wild because Amani's like the real influencer. So like, what is it when I influence the influencer?
Maybe in a more analog way. We all influence in our own ways.
Analog influencers making it happen.
It's the new thing. I want to hear from all 3 of you. What does like a regular day in your lab, in your life as a researcher look like to you?
Gabi,
I can assign the first person to go if that makes it easier.
Yeah, you go first,
Gabi,
Gabi.
let's hear from you.
Okay, so I'm still an undergraduate student. So now that I'm away from the main campus in Boca, I'm not doing the hands-on taking SEM imaging. But when I was on main campus, I would receive the dissected lamellae samples from Lauren, and then we'd go through the whole process of first drying them with a critical point dryer, then sputter coating them with a gold palladium filament, and then just spending like hours behind a scanning electron microscope and taking at first like overall images of each lamellae, and a lot of times you have to take a few images and then kind of line them up, and then whenever you see something kind of extra cool, you know, like a region of spaghetti or whatever, kind of zooming in and taking even closer up pictures with an SEM of those regions. But yeah, now that I'm at the Harbor Branch location, I am kind of just evaluating the images I have taken and making note of different regions we've seen with sensory, non-sensory tissue there. So yeah,
The like
not as
trained
cool.
eye.
Yeah.
But yeah, no, totally. That's awesome. The trained eye that you have to like identify a interesting spaghetti region. It's really, really excellent to hear.
No, it's like I'd ask Lauren about a million questions when I first started, and I still do now. And talk about trained eye, like hers is crazy. It's really something. So, yeah.
Let's see, Amani, how about you? What does an average day in the lab look like for you?
In the lab, I do a lot of CT scanning, which can be hours at a time. I've done like 8-hour days next to a CT scanner being a little gremlin. Other times I spend all of my time on the computer looking at the CT scans that I did and building things and yelling at 3D printers that won't print things the way I want them to.
It's so real.
You see that I have like
Yeah, like everybody has said, every day is kind of a different day. There's definitely some days that I'm waking up earlier than I want to get on a boat and catch sharks and bring them into the lab and do some experiments. There's days that I'm driving to get shark samples from somebody that was like, hey, a shark washed up and we have the head, or me begging fishermen when they catch sharks, be like, can I have the head, please? Like, if you're going to kill the shark, at least take the head. But then there's days where I spend a whole day on the computer or at the microscope, which is my happy place. And like I said, I work in a unique K through 12 environment as well. That's where my lab is actually housed. So there's days that I am with kindergartners and I'm talking to them about how to use a scanning electron microscope, which is crazy. Or we're CT scanning their toys to show them, like, this is a toy lizard. This is a fake lizard. This is a real lizard. This is how you can tell what real versus fake is. So it's a lot of education and research and mentoring, a lot of different stuff.
Oh my gosh,
I'm
my
jealous of those
kindergarten brain.
kindergartners. Where was that?
Yeah.
When I was,
Yeah.
We're all going back to kindergarten.
Yeah, still there, Lauren's class.
Dr. Lauren,
Please,
yeah.
Dr. Lauren, teach us about real and not real things with a scanning electron microscope. That's amazing.
Yeah.
And finally, this is something that we got to return to from what Amani said earlier. The nurse shark debacle. I'm sorry. What's the deal with nurse sharks?
Lauren's head says it all. So
What's the
nurse
beef?
shark. Nurse sharks, I love them. If you think of what a nurse shark does all day, it sits on the seafloor. And unlike something like a great hammerhead that swims all of the time, the nurse shark has to be able to get off of the seafloor, which means that it has specialized muscles that allow it to push itself up and off of the seafloor when it's trying to get up to go swimming, either for prey or to just move 5 feet because they're lazy. So we catch nurse sharks and we tag them and take other morphological data. We measure them, we draw blood from them for a lot of different types of studies, and then we release them. And getting them onto the boat is often called a nurse shark rodeo because you will pull them onto this platform that we have that sits off the back of the boat. And because they can push up, they'll do like push-ups against you and they can spin themselves in all of these different ways. And it's mostly just you trying to leverage your body to get it to just sit. And unlike other sharks, so big sharks, especially something like a bull shark, we pull them up, and because they don't really have a predator, they just sit there and we do the thing and then we let them go. And they're like, cool, I sat out of the water for 2 seconds. It's great. Nurse sharks, no matter what time of day it is, no matter how long they've been out of the water, even if it is literally 5 seconds, they will just be mad at you the entire time and just like spewing nurse shark hate your way and angrily sucking at you because they suction. And it's a whole thing. So this is the rage that nurse sharks unleash on the world when they are brought above the water.
Yeah, they're great at like alligator death rolls. Like that's literally what they, like, as you're pulling them out of the water, they're like a tornado. And then when you actually do have them, like a lot of times sharks, they have nice jaws. So you can grab them really, like you can secure a shark pretty nicely behind their jaws. But like you know, like when you see people that work out a lot and they just have like a neck, that's what a nurse shark is like. Like they have no head. It's just one neck. So you're trying to hold it and it's just like twirling. There's nowhere to grab. And then they also hurt because their skin is so tough. It's like the toughest. And it's just a struggle.
Yeah. From dermal denticles.
From the dermal denticles, which you can actually see with your naked eye on a nurse shark because they are that large and they are also that tough.
What?
So everyone is always like, oh, nurse sharks, they're so sweet. They're like little puppies. And I'm like, rage. Nurse sharks are not sweet. They are not puppies. They are the angry lion that needs food.
And they were your gateway shark.
I get it now.
Yeah, listen, I love them. It's okay. They can go to therapy one day. It's fine.
Toxic.
Well, thank you all so much for the laughs, for the knowledge, for these talks. This has been an amazing program. I'm going to send you all to the backstage area. But we truly so appreciate you all being here and sharing with us tonight. Thank you so much.
Thank you.
Well, thank you guys.
Yeah, thanks, guys.
Good night.
We're back, and I'm gonna say thank you again, because literally not enough thanks for the delightful.
Thanks are due.
Thanks are so due. Amani, Gabi, Dr. Lauren, it's been a delight. Thank you for showing us the incredibly dazzling place that is the inside of a shark's nose.
And our audience, we would just love to remind you that Night School is live the first Tuesday of every month. And now that's the first Tuesday in 2024. With insightful and interactive conversations with scientists and their friends. So that means that we'll be back on March 5th with a program that'll take us around the world and into the lives of scientists who conduct fieldwork in some of the most remote, stunning, and story-filled places on Earth, all to learn how birds change with their environment and how the environment changes birds.
Very excited about it. And until we do return, you can rewatch past episodes, which are now 93, including this one, on our YouTube channel.
And if you'd like, I threw up a QR code. It's there. It'll send you to a link. The same link is going into the chat. It's a survey. It'll help us to continue making Night School with you in mind. Regulars and newcomers alike, we'd love it if you took a second to fill it out. For those of you who have already filled it out in past episodes, we love you. You're the reason we do what we do. I would be crying every morning when I wake up without you. And that's not hyperbole. I'm very sensitive.
You heard it here first. Thank you again for joining us. We will see you all March 5th. Have a great night.
Good night.